This short video provides a brief summary of my project on studying influence of pH and acidic side chain charges on the behavior of designed model peptides. The biological membrane is heterogeneous, crowded by a variety of lipids of various lengths and by a variety of proteins. This brings in a lot of complexity for experiments to examine and understand membrane proteins in detail. The lipid bilayer has been shown to alter protein shape and function, like for example, rhodopsin's photochemical functions as well as the enzymatic activity of cytochrome oxidase C are affected by the bilayer curvature, thickness, and elasticity. The transmembrane protein segments are typically composed of a hydrophobic core, with their sequences determining their function. Despite the predominantly hydrophobic core, potentially charged residues are also observed somewhat often within the core segments of a transmembrane protein and are believed to be important in the functioning of many of these proteins. These titrable protein side chains, notably those of aspartic acid, glutamic acid, histidine, lysine, and arginine, encounter an environment-dependent energy barrier that governs their ability to pick up or lose a proton in transitioning between charged and neutral states. The charge status in turn will influence the helix orientation and geometry in bilayer membranes. Dynamic transmembrane helix motions are crucial for the functions of the membrane proteins, and subtle molecular interactions can govern the stability of native states and biological activation of these membrane proteins. Voltage-gated channels like potassium, calcium, and sodium channels play crucial roles in excitable cells. Each of these channels contains voltage sensors and a selective ion conduction port, with the general structure comprised either of 4 independent protein subunits or one long polypeptide containing 4 homologous domains. Each domain contains 6 transmembrane segments S1 to S6 and a pore loop between segments S5 and S6. The segments S1 to S4 form the voltage sensor. Of key importance is the S4 helix, which was recognized as a voltage sensing unit owing to the presence of basic residues, arginine or lysine, at every third position. This segment, along with the S2 helix, is rich in intracellular acidic residue, are involved in gating. Charge residues have also been implicated in aiding fold switching of metamorphic transmembrane proteins like the chloride ion channel CLIC1. The CLIC1 intracellular chloride channel exists in both the soluble conformation in cytoplasm as well as membrane bond conformation. This conformation is dictated by the pH sensitivity that is governed by specific residues functioning as pH sensors. The monomeric soluble protein has an end domain comprising of helices alpha 1 and alpha 3 and a beta strand B2. The alpha 1 helix and the B2 strand form the transmembrane domain. The conformational switch results when the end domain detaches itself from the C domain and restructures itself for insertion into the membrane. In the cytoplasm, at a pH around 7, glutamic acid 81 on alpha 3 is charged and forms a salt bridge with arginine 29 on alpha 1, which stabilizes the native conformation. As CLIC1 nears the low pH membrane surface, Glu 81 loses its charge, causing a break in the salt bridge. This allows the detachment and insertion of the end domain into the membrane. Addressing how molecular changes in the protein influences their behavior in membrane environment requires significant computational and experimental studies using model systems. However, membrane proteins typically consist of multiple spans through the bilayer that lead to protein-protein interactions and forces that create more factors, complicating pure protein-lipid interaction studies. So to simplify this problem, we make use of small alpha helical peptides with a single membrane span. We make use of GWAL-23 model peptide system that represents a typical transmembrane helix comprising of a core of leucine and alanine repeats with tryptophan or tyrosine residues anchoring them. The N and the C terminal are capped to ensure that the peptide is neutral. The peptides have relatively low dynamics on the NMR time scale and are sensitive to changes in the membrane environment. We use this peptide framework to incorporate glutamic acids in the core at positions 14, 16, and 12. At position 14, glutamic acid residue is slightly off-center and lies on the opposite phase of the two tryptophans. At position 16, the glue residue is quite close to the interfacial layer but is located right below one of the bulky tryptophan side chains. The blue residue, located at position 12, is placed at the center of the peptide, right between the two tryptophan cages. Apart from the glutamic acid, we also incorporate a pair of deuterium-labeled alanine in the two of the core positions, either at 7, 9, 11, 13, 15, or 17, and at times towards the helix ends as well, near alanine 3 and 21. These labels help in analyzing the peptide orientation and dynamics using solid-state deuterium NMR spectroscopy. The alanine pairs are labeled at 50% and 100% abundancy levels to give different intensity signals. Now, solid-state NMR differs from solution NMR, where in solution NMR, molecules reorient fast and randomly, giving singlet peaks of chemical shifts for each molecule. Solution NMR are much suited to study the internal structures of peptides. In solid-state NMR, however, orientation of the molecules are fixed relative to the magnetic field. The deuterium nuclei used for analysis is a quadrupole, and hence gives a split peak. The distance between the two peaks is measured as a quadrupolar splitting. Once we gather these data points for 6 alanine core labels, we can use them in our calculation of geometric analysis of labeled alanines, or a method called GALA, which helps us calculate the tilt and the rotation factor. This tilt and rotation factor is called theta. Theta, as I mentioned before, is a function of the tilt and rotation of the peptide. The tilt of the peptide is with respect to the membrane normal, and the rotation of the peptide is defined as the direction of the tilt. Using average peptide orientation, we can investigate incorporation of different residues by monitoring the changes in reorientation. We prepare our samples by applying a peptide-lipid mixtures to approximately 40 glass slides and dry them down. We then hydrate them in excess water environment of 45% weight-to-weight buffers that are prepared in deuterium-depleted water. We then stack these slides on each other and place them inside glass cuvettes and incubate them for 48 hours at 50 degrees centigrade. The mechanical pressure and the surrounding environment allows the lipids to form a bilayer, and the peptides orient themselves into these bilayers. The samples are then placed in NMR probes at an angle that is either parallel or perpendicular to the magnetic field. That is called beta 0 or beta 90 respectively. The metal side chains of the deuterated alanines have fast rotation in the magnetic field, and hence the quadrupolar splitting measured at beta 90 is half of that at beta 0. We investigated our peptide behavior in synthetic 12-carbon lipid bilayer systems called DLPC, or dilaryl glycerophosphocholine. These are thin lipid bilayers. Our first investigation was with glutamic acid incorporation at position 14. Our pH-based experiments showed that glutamic acid incorporated peptides show no changes in the peptide orientation between pH 6 to pH 12.5. but showed changes at pH 13, suggesting deprotonation of the glutamic acid side chain at a pK around 12.5. We observe a 6-degree change in the tilt of the peptide between the neutral and the charged helix, along with changes in the helix unwinding towards the C-terminal. We observe the core helix to be interrupted or unwound from residue 17. This result was quite unique for glutamic acid. Deviations from the core helix geometry have not been observed for arginine, histidine, or lysine at the same position. The glutamic acid positioned at residue 16 in the core helix showed titration behavior not far from glutamic acid 14. But what is unique about glutamic acid at position 16 is the extent of helix unwinding near the guest glutamic acid residue between pH 6 and 13. The alanine 17 residue was found to be unwound in E14 peptide, is found to remain part of the core helix in GWAP 23E16 peptides between a pH range of 6 to 12. At pH 13, however, both residues 15 and 17 that surround the blue 16 residue become unwound. Due to significant unwinding of the C-terminal at pH of 13, the remaining helical core of the peptide shifts upwards, straightening the peptide to allow access for the charged glutamic acid to the interfacial layer. For the case of GWAP 23E12, When the pH is raised from 6 to 13 in the LPC ether biolayers, there are no observed changes in the peptide orientation, and furthermore, we don't observe any changes in helix frame either. This suggests that the glutamic acid residue does not titrate at this position, even at a higher pH, and may have a pKa that's higher than 13. Our combined results suggest glutamic acid residues to have significantly high pKa in hydrophobic bilayer membrane environment. The change in helix terminal unwinding is a unique feature observed in glue-incorporated peptides. There is a position-dependent change in the pKa and the unwinding change that together govern the peptide stability in the membrane bilayer environment as the glutamic acid titrates. Thank you.